Okay, so we're going to move away from microscopy, and now we're going to look at biophysics. And the facility here is just me and Chris. You see Chris over there. And we're available for advice, training, collaboration, and experiments. And also we run the Next Gen Biophysics Symposium in September, one day. Exciting symposium. So in biophysics, the kind of questions that we kind of get asked are all these kind of different ones. And being students, and obviously scientists in the LMB, you're very, very blessed to be here because we have got lots of different techniques to be able to answer these kind of questions. So today, then, I will basically be talking about fluorescence and mainly about bulk fluorescence. Kristen, in the next lecture, will talk about single molecule frets and optical tweezers. And also at the end, then I'll talk about microscale thermophoresis and affinity. Okay, so first of all, we're going to kind of describe the phenomenon of fluorescence so you can understand why it's used and what the pitfalls are as well. We'll look at some applications, and in the end, we'll just do a special application, which is microscale thermophoresis. Of course, so we're looking at fluorescence, so light's interacting with matter, and it's quite curious if you kind of think about it, that actually light kind of will interact then in the visible region then with electronic transitions in the outer shell or ultraviolet the inner shell. And we get this process of fluorescence with aromatic compounds in pi-to-pi star bonds, and also in the lanthanides as well. You can actually get fluorescence as well. All right. So the kind of the groups that you get, you can kind of see they're all kind of ring structures, and that kind of tells you about about something with the electronic structure. Including say GFP down here at the bottom, where the kind of the ring is then the fluorescence group is then formed by conjugation of the amino acids, and different forms of GFP that have different substituents, different amino acids, which kind of change how the electrons are distributed across this ring system. And then in the corner there is a synthetic fluorophore. So what happens in fluorescence? Well, you must have some light getting in. Here's the Jablonski diagram, ground state, S1 is S2, higher energy levels. So you get an excited state when light interacts. And then you go up to the upper level, but just by thermal fluctuations, etc., vibration levels, then this kind of heat's dissipated really, really quickly. But then, so you're in this kind of upper level, and then after about another second, you'll then get transition down, and of course, it's losing energy, and then the energy is given out by light. And I think you can probably see then that actually the ground state might have different vibrational levels, and so you're going to get a spectrum that depends upon that electronic structure. So then what you tend to get then is you can see that the emission is then over in the red here in a big kind of spread of different wavelengths coming off, because then the energy is related to then the wavelength. And you also then, you see you're getting excited by a shorter wavelength, and that's got higher energy, but you only get back less energy through longer wavelength. And this kind of distribution is called a Stokes shift after Gabriel Stokes, who was here in Cambridge. Is there anybody from Pembroke? No? Anybody know where Pembroke is? Well, next time you're kind of in town, go into Pembroke Chapel, have a look, and there's actually a stained glass window to Gabriel Stokes, which is not a bad thing to have, right? So the important thing, of course, is that because you're doing an excitation, it doesn't really matter what level you kind of go to. So you can actually then put more energy in to get up to a higher level, but the fluorescence is really going to be the same. So when you got a fluorophore that has the emission and the excitation quite close together, you can actually excite at a much shorter wavelength, and you still get fluorescence. And you'll try and avoid getting that kind of bleed through of the incident light. Right. Okay, not everything goes up, so you can get fluorescence may not come out. So you may get quenching, or you may get energy transfer by fret. We've seen in the storm that you can get bleaching, and of course, in storm microscopy, that's very, very useful to get bleaching. But in our kind of systems, getting bleaching, if you're doing single molecule work, isn't great. And if you get rearrangements, then you get phosphorescence. So how good are fluorophores? Well, we can kind of say fluorophores are good by the amount of light that comes in, the number of quanta that comes in against the number that come out. And that kind of reflects the kinetics of the fluorescence against these kind of non-radiative processes. And that can depend on the solvent and on the exposure And also not all fluorophores are equally as bright. So given all those kind of things about fluorescence, we kind of use it. It's energy interacting with matter. It's very, very sensitive to the changes in the environment. That's a good thing, but also can be a bad thing if you don't do good controls. It's got a very, very fast response time. It's very sensitive, down to picomolar. It's very selective if you've got a group or an extrinsic fluorophore. And it's also reproducible unless you have bleaching. Right. So let's kind of look at intrinsic protein fluorescence. Okay. You all know this is kind of tryptophan, phenylalanine, and tyrosine. They all absorb. One good kind of tip is that if you excite beyond 295, you'll really mainly hit the tryptophans. So that'll be quite good. We'll see later when we're looking at nucleic acid binding. Phenylalanine has got a very, very low quantum efficiency, so it doesn't tend to ever be involved in the fluorescence. And you can see both tryptophan and tyrosine then have got in water then have got slightly different emission spectra, right? Of course, where is that kind of maximum on the spectra is really depends upon the environment. So if you have something in if you can move from a hydrophobic, say the interior of a protein, out into the solvent, you're then going to get a red shift in the maximum. Other factors that kind of affect the fluorescence is temperature, which we'll see later with the Prometheus, pH, and also in kind of old school, people used to do solvent accessibility using quenchers such as acrylamide and iodine. Right, so let's look at some questions and kind of techniques, what's kind of good for. So it kind of really highlights the sensitivity and the reproducibility of using fluorescence. We're going to go back into protein folding. And so this is using nature proteins with these small characters at the top, urea and glutathione. And of course, the unfolding then leads to the exposure of the fluorescence groups. And at the bottom here, you can kind of see from zero glutathione then in red, you get a massive increase in both the light given off, and also there's a shift as well. So if we just took one wavelength, then we could follow the unfolding process with increasing amounts of denaturation. And you can see that what happens is, in this kind of flat region here at low concentrations, it's all folded. High concentrations of denaturant, then everything's unfolded. And in this gray box, then what you've got not just partially unfolded or all folded. It's either folded or unfolded. This is a two-state folding process. There's only either an unfolded top there, an equilibrium between native or unfolded. So we can relate to that. We're just going to go through how we would actually fit this data. Don't worry about the details. So we've got an equilibrium. And we've got two baselines here. We've got a native baseline of fluorescence, just a linear function for the unfolded. And so in the middle, that fluorescence depends upon the fraction that's folded and the fraction that's unfolded, given what the native and the unfolded fluorescence is. So of course, if we know what the fractions are, we can work out into the binding constant, or sorry, the equilibrium constant, and for each of them. And of course, that's linked to the energy. So the free energy of folding is linked to this binding constant, natural log, times the gas constant, times the temperature in Kelvin. But of course, you need to have and then we can rearrange that. It's an exponential function. And people have found then that there's a linear relationship between the denaturation of the free energy, standard free energy, and the denaturant concentration with this M value here. So that when you've got an equilibrium, when everything is 50-50, then that's a delta G, or that's a K value of 1, natural log of 1, 0. So then the energy is 0. So we can then say that at this delta, the standard delta G is then equal to the concentration of denaturant where 50% is folded and 50% is unfolded. So we can just then go through the maths. Don't really worry about that. The important thing is that we're just measuring the fluorescence, from the fluorescence, we know what the denaturation concentration is, and then we work through, and then from this, we can then work out the stability of the protein. And of course, why we want to do that? Well, in this study here from the Isaki lab down in pharmacology, they looked at point mutants in the BRCT domains in BRCA1, and to try and understand what they were doing. So you can see if you go into the top right here, you can see that, well, if these mutations stabilize, then relative to the wild type, then you're going to have a higher barrier to go across, and a much bigger gap in energy between the unfolded and the denatured. The native and the unfolded. And so there's going to be more of the protein then folded. If you destabilize the native form, then there's going to be much more kind of going over that barrier, and so you're going to have much more unfolded. And this is what they see in the bottom here. Lots of them are destabilizing. Some of them have no effect at all, and some of them are actually stabilizing. So of course, the important thing is that the destabilizing mutants in body temperature, that's maybe going to have more unfolded material of these BRCT domains, which then means it's not going to work. Right, so another way of looking at the stability is looking at thermal unfolding and then following the fluorescence. So again, you can imagine you heat something up, it unfolds. But you can use this then for screening. If you said you had something that bound on to the folded material, you're going to have another equilibrium there. And then it's going to shift everything across. So you're going to have an increase in the melting temperature. And we've got a lovely instrument downstairs, which unfortunately is broken, but it's being repaired. And hopefully we'll be back soon. So don't quite rush to go into it just yet. So it's called Prometheus, and it's nice because it's computer based. You can do 48 samples, very small samples. It's got very accurate temperature control. It's got a large dynamic range. It's using intrinsic protein fluorescence. So whether your protein works or not depends upon whether it's got fluoric groups and whether and what the fluorescence of that. You can use membrane proteins as detergent, and you can also do backscattering. So again, here's another protein. We're going to unfold it, and in this case, then going from the native to the unfolded, we're going to have a decrease in fluorescence. So if we follow it by temperature, you can see one problem with kind of doing the analysis this way is that if you look at the unfolded, the fluorescence goes down. And similarly, with the folded material in blue, it goes down, and then you get this transition, which is where it's unfolding. So it's quite difficult to follow. So what you can do to get over that is then actually look at two wavelengths and then look at their ratio. So you can see here, they're both kind of going down similarly. And if we look at the ratio, then in the middle there of 350, 330, you can see you get a transition. And then doing the first derivative of that, you can get the inflection point, and that's kind of a TM. So I will say a word of warning, which is when you kind of these are quoted by TM, it's not, the protein may not be in thermodynamic equilibrium, so therefore it's not actually a TM. Because lots of the times we'll see in a minute, you get aggregation, and so you've got another component. Right. But what you can do, of course, is that you don't have to really worry about getting that it's not a thermodynamic TM. You can look at then, say, different buffer concentrations, or and see when your protein is more stable. In this case here, you can see that putting in 10% DMSO is a good idea. The instrument also has this kind of backscatter, so that what you can do is look at, do you get any aggregation or not? Okay, so any aggregation or not. So if you get big aggregates, you get light scattering. So the light that's bouncing off the bottom mirror will get less. Chris will talk about this in depth, so I'm going to go into it now. Here's an example from the James group. We kind of used this very well. So HIV, they made the HIV capsid, and then when they saw the structure, you could see that they found in the capsid there was this pore formed by then this ring here of 6 members, and it was in open and closed. And so they thought, well, could we have a drug, or could we stabilize this? What would happen? And they used then different levels of these substituted benzenes, all with different carboxyl groups on there, because the ring itself was quite positively charged. So if you look down at the bottom, blue, and then going across, and then you're getting more and more substituents here, you can see that the melting temperature is going all the way up. So then they're locking in all these subunits, and you're getting much more stability. Another way of looking at binding is something that kind of tends to be forgotten really in terms of kind of labeling and everything, is that sometimes you can actually look at just the intrinsic protein fluorescence of a protein. In this case, this was a ATP, as it's a chaperone, and ATP nucleotide would bind into the pocket there. There wasn't any there wasn't any tryptophans or tyrosines actually in the pocket, but there was a conformational change which then caused a quenching of the fluorescence. Because then we're looking at nucleotide, then I excited at 295 because obviously nucleotide absorbs at 260, so you don't want all the light to be absorbed by the nucleotide going in. And so what you can do then, if you titrate in a ligand, in this case, it was a non-hydrolyzable nucleotide, AMP, PMP, and then you can then titrate then the change in the fluorescence and then get a binding curve. So in this equilibrium here, we are then assuming that the change in the fluorescence is actually comparable to the amount of complex we find. So we want to then work out this KD from the free protein, the free ligand over the amount of complex we formed. Okay, so but what's going to happen is, of course, that the amount of free protein is going to be reduced by the amount of complex we're forming. But what about the ligand? Well, we assume that because the concentration of because the concentration of the protein is quite low and the concentration of the ligand was really quite high, then really the amount of ligand you're putting in is essentially the same as the free ligand. And our assumption we're going to make here is that then that the complex is related to the change in fluorescence, and then when you get to the maximum, that's going to be the total protein. Just a simple formula, and then you can just fit that by the change in fluorescence and the ligand binding. Of course, then you can use fluorescence then to look at pre-steady state kinetics. So if you want to know then the individual on and off rates, you can use stop flow, which we've got one here. So you've got two syringes here, one with protein, one with ligand, and they're going to be driven at very high speed straight into this mixer, through into the chamber where you can do absorbance or fluorescence. And because there's a whole column of liquid, when you push something in, then the liquid is going to be pushed out, and let's go on to this stop syringe here. And when that gets pushed out, it hits the trigger, and then it goes and starts recording the fluorescence change. And here you can get down into the millisecond time frame and then look fitting and then fit the absorbed rates against the concentration of ligand. But lots of people use extrinsic fluorophores. So another way of, say, looking at protein stability is using this dye called Cipro-orange. So it excites and emits there, and then when it binds to hydrophobic regions that are exposed when you have a denatured protein, you get a big change in fluorescence. And of course, the dye was chosen because it's comparable to the filters in a qPCR or an RT PCR. So you can then put this into a PCR and different conditions. So the kind of the data you do get out is a bit sloopy. Again, because the fluorescence is actually temperature dependent, but also at high concentrations you might be getting more aggregation, so you might get a change in then the binding. Another way of using a extrinsic fluorophore is by, say, using it as a reporter. In this case, we've got a phosphate binding protein, which has been labeled with a coumarin derivative, and when it binds inorganic phosphate, it then has a big change in the fluorescence because there's just a slight domain movement. And then we can correlate that against a standard curve here of inorganic phosphate against the change in fluorescence. And so we can then look at the mechanical kinetics of this enzyme that's cleaving ATP and then leaving out PI, measure the change in fluorescence against time, and then fit the initial rates and convert that into phosphate. Okay, let's move back from then binding or back to binding again. So if we want to look at interactions, we can use anisotropy and polarization. This tends to be quite a common method to do because it uses very, very low amounts of labeled material. And lots of processes, then you're getting changes in mass, either by things binding to each other or, say, things being cleaved off. Usually we will then label the smaller of the of the components. We'll just go through just how this works. So then you can again avoid the pitfalls that people fall into. So let's imagine then we got light shining onto a fluorophore. It's going through a polarizer, so it's going to go in one direction. Now, the light will only be absorbed if that dipole of that fluorophore is in the right direction compared to the light. It absorbs. Now, free fluorophore by itself is going to tumble very, very fast. So if you remember, then it's about 1 nanosecond for this fluorophore to emit. So then when it emits, it could be in a completely different orientation. But if, say, then we start to bind something onto there, we're getting a change in mass. This is then going to rotate much, much, much more slowly. So then by the time the fluorophore then comes to emit its light, then it could really be in the same orientation that we started off. So we could say here that the polarization has been maintained. Well, in the previous case, then it's been here, it's kind of being lost. You got depolarization. So how we measure this tends to be either in two formats. You can either have it in T format, where you've got two polarizers, or a lot of times we'll have it in just an L format, where you have a polarizer at the start, and then on the incident light to make the polarized light, and then on the detection, then you have a polarizer that can actually shift vertically or horizontally. But it's easier here, I think, just to kind of think about it in terms of in an L format, sorry, in a T format. So, okay, so imagine what's happening is that, right, when you're starting to get, let me go back again. Right. So initially, then you've got just a fluorophore or a small molecule kind of that's fluorescing, or a small protein that's fluorescing, and it's tumbling quite fast. So then when you look at the light's coming off, it's going to be depolarized, and so you're then going to get a kind of a reduction in the light that's with the same polarization as the incident light, and you're going to have maybe more kind of coming through in the horizontal. And then when we slow things down by binding the tumbling down, we're then going to get more kind of polarized light in the vertical direction. So if we titrate in, say, the protein, then what will happen is then you can see the vertical polarized light coming off increases, while that in the horizontal in blue then decreases. So polarization, or more correctly, kind of anisotropy, is really just a difference between these two light intensities at the vertical and the horizontal over the total amount of light. So it's just a ratio. Also, coded will be a lot, you'll see in papers and everything, is polarization, which is slightly different because it's got a different, there we go. And the reason for that is that it's not, polarization isn't actually directly proportional to the total amount of incident light. So the anisotropy is the one to use because that's got twice the amount of light there. It takes into account all the different vectors. But it's easy just to convert in papers and stuff. You can just convert from polarization to anisotropy. Also, you have to take into account if you're using a perimeter, the grading factor. So the actual polarizers may not be necessarily perfect. So you might have to adjust the intensity of the light. And that you can just do automatically, and the plate reader does it for yourself. So it's really important also that you calibrate at the appropriate, what you're expected in the plate reader at the expected value that you're getting. The default one in the plate reader is just used for small molecules, so it has a 35 milli polarization units. But your protein may be 120. So you must make sure you put in the right value to do the calibration. Right. So what fluorophore do you actually choose to kind of do polarization? Well, it depends upon the mass you've got. You can almost kind of think about it. It's a polarization will happen then when you will depend upon the lifetime of the fluorophore against the tumbling rate as well. So the Perrin equation at the top kind of shows that. And this tumbling then say, a protein will depend upon the viscosity and the volume of the protein, gas constant, temperature in Kelvin. And this volume then depends upon the mass of the protein. And then this is the particle specific volume and hydration level. So down at the bottom, we can see that different lifetimes then will give you different readings depending upon what molecular weight you have. So you can see that if, say, you had a large system, then in red, and you had a very, very short-lived fluorophore attached, it's going to go maximum initially. And say, so you may then need another fluorophore with a different lifetime. And of course, that's all in the literature and on databases. Right, so let's use polarization as we often do for binding. So in this case, we've got a very, very low concentration of labeled ligand. It's kind of because we're looking at a lot of the times we're looking at tight binding. It may be equal or just slightly below the KD. And then the data, we're going to start off with the baseline, which has then got the initial polarization or anisotropy of the system of the protein labeled. And then you're going to add in the binding partner. And then in the middle, that's going to depend upon, obviously, the start. And then the relative change in the polarization depends upon how much of the labeled material is actually in the complex. Again, we're going to go through, again, for the MAVs. So this time, because the concentration of the ligand is so small, and we're titrating in, and it's kind of close to the KD, we can't say then that the free protein here, or the total protein here we're putting in is equal to the free protein. So we then must take away then the amount of complex we're actually forming from each of the free protein to get the free protein. And of course, how do you solve that? Well, it's just really a simple quadratic equation down at the bottom here. And so then that's just going to relate the fluorescence change, sorry, the polarization change, initial polarization, the change that you actually see, and then the total, you can put in the total protein and then the total ligand that you've got, and then that will fit. So this equation is called a ligand depletion equation, because essentially as you're titrating in the binding partner, you're reducing the amount of ligand and you're reducing the amount of free protein as well. So here's a really nice example of how you can kind of use polarization and how it relates to mass. So this was checkpoint proteins in the Barford lab with Elise Fisher. So they had this protein, part of this protein MAD2. It's a C-terminal domain. Now it was basically a homodimer, and it had a coil at the bottom. So they thought, they knew that the BUB1 and the CDC20N were involved in forming a complex. So what kind of complex did they form? Was it just BUB1 binding, or was it CD20 binding, or was it kind of a mixture of the both? Okay, we need to solve this with polarization. So if we have labeled then BUB1, titrated MAD2, okay, we're getting a nice change in the polarization. We can kind of fit that. And then we can look at changes in phosphorylation, whether that affects things. So then when we come to then titrate in the CDC20, if say, either of them could bind, there was kind of a competition for both sites, then as you titrate in the CDC20, you expect then the MADs to be, the BUB1 to be displaced, and so you get a decrease in the polarization. But that's not what we saw. We saw an increase. It meant that the model actually had to be this. So there was actually, even though it was a homodimeric protein, the MAD2 had asymmetric sites. And we can show that again in another way by then just having, if we have then the label BUB, and then titrate a preformed complex of MAD and CDC20. And then you end up, again, starting off at the same time, or if it's slightly higher because it's slightly a different size. And you get up to the same endpoint polarization. So you're forming the same complex with the same mass, which has the same tumbling, which therefore has the same anisotropy value. Okay, so Another way of looking at complexes is using fret. So in this case, what we're going to get, we're not going to excite one protein. In this case, imagine it's kind of blue fluorescence protein, and it's not going to then fluoresce. What's it going to do is it's going to transfer its energy to the GFP, and then the GFP is then going to then emit the light. And this is because there's dipole-dipole interactions between these two proteins. And the energy transfer really is dependent upon the amount that's stored by the donor, that's the one you excite, against the quanta transferred to the acceptor, which is the one then obviously that gives off the fluorescence. And that's related to the transfer time and then the lifetime of the donor. And then, so this is, we can figure that out. And fret only occurs for certain fluorophore pairs. They need to have an overlap then between the emission of the donor and the excitation spectrum of the acceptor. And if we look at FP base, you can kind of see lots of different ones. And why do people like to do it? Well, it's really sensitive to distance between the two fluorophores. So that, and it's the power of R to the 6, the distance. And so each pair of donors and acceptors have a specific R naught here, where you get 50% of the transfer here. And then that depends upon coupling constants and reflection index and orientation. But typically you're looking at between 20 and 70 angstroms. So you can actually use these as molecular rulers to be able to look at distances between things. Or alternatively, you could use it for, say, binding of two things together if you have different fluorophores, or you could look at conformational changes. When you come to do an experiment, it gets a bit more complicated because not all the energy is transferred. So we're going to excite the donor. We can see we're going to get in here, we get the donor emission, and then some of the energy is transferred, and then we get the acceptor emission as well. So how do we actually calculate it? Well, if we do it from donor emission, that's much easier, but we need to then do some controls as well, where we have then just the donor by itself. So then we can see how much energy is actually lost then when we have the acceptor in here. So then the energy is just this. It's just a 1 minus then the fluorescence in the presence of the acceptor over the in the absence of the acceptor, and then you need to factor in the amount of labeling you have if it's labeled. If you do it from the acceptor side, then this becomes a bit more complicated, because you might then have fluorescence of the donor at the acceptor wavelength, and also with the acceptor, even though you're exciting at the donor fluorescence, which is far away from its normal thing, you may get a little bit of fluorescence coming off, but that's it's only a little, but it's still significant. So you need to take account of that. And then we end up down here at the bottom, where the energy transfer is really dependent upon the ratio of the presence of the acceptor in the presence of the donor over its absence, and then related to the extinction coefficient of the donor and the acceptor at the wavelength of the excitation. Over again, how much you've kind of labeled if you've got a label system. So here's an example from the Chin group. So this was an example for them to show that when they put unnatural amino acids in, they could actually form them. And the non-natural amino acids they put into calmodulin here were then reactive against different dyes. And they could show that then by unfolding the calmodulin then with urea. So again, as you see, if you increase the urea concentration, then what happens is that you reduce the fret because they're becoming farther apart, and you're getting an increase in the donor fluorescence and a decrease in the acceptor fluorescence. Okay, we've got lots of different plate readers and perimeters here, and obviously, with good old perimeters, then you need larger volumes, but they're very, very flexible. You can change everything. While with our plate-based readers, they tend to then be fixed. We've got optical modules, So we have fixed wavelengths that we can actually use. But if you look at the biophysics website, you can see which ones we've got. Okay, practical tips. Filter everything, spin things down, check that you have things aren't aggregating, make sure everything's clean, concentration's accurate. Make sure you're not getting sticking to plates or to the cuvettes. Make sure you've got good signals. Have you got the right settings? I kind of talked about the fluorescence, the plate reader for polarization. Are you getting photo bleaching, background, inner filter effect I didn't talk about, and make sure you're doing accurate concentration for your titrations. Right. Additional controls, these are quite important. If you've got something labeled and you're looking at binding, then a binding partner, make sure that the binding is to the protein and not actually to the fluorophore. Of course, you can do that easily by just having a free fluorophore and your binding partner. Or alternative, what you can do is say if you had, say, a small molecule that was binding in, that was a fluorescence reporter, you could then look at displacement out. And temperature controls, you can see fluorescence can change with temperature, so that's very important. And also all these kind of binding experiments, the time to equilibrium is quite important. After the experiment, data fitting, we'll talk about that in a future one. Okay, so the last 15 minutes or maybe less before tea, given the fact that we were rudely interrupted, we'll talk about MST. But so we've got a very new machine downstairs. It's called the Microscale Thermophoresis. We call it MST. And this is a process that you're kind of all aware of electrophoretic mobility. So thermophoresis is really a kind of movement in a temperature gradient. And that will depend upon the charge and the size and hydration shell. So here we go. Let's see if these will actually work. And there we go. So we've got an infrared laser going all the way through these beads here, and you can see that it's DNA on the left-hand side there, and you can see that when this goes through, and then on the right here we've got microbeads, and then we hit the spot here, then we get movement in, and then this one, then you've got the laser kind of going down, and then the DNA is kind of moving out. Of course, it was developed then by Stefan Durr in, unsurprisingly, the LMU. I think it was during his PhD. So as a physicist, he worked out then how this process worked. So really, you're getting a concentration difference between the hot and the cold. And this depends upon this constant here and the change in temperature. And then he found that what really depended upon was the size, the hydration shell around the protein, and then the charge. So the format of the instrument is little capillaries, like the Prometheus, and then you've got you're looking down from the top there, and you've got a capillary filled with fluorescence literally protein or anything else, and with a binding partner, and that fires an IR laser down, and also then it's firing down a fluorescence detection system as well, and you're getting a very, very concentrated spot of heat. And the dyes that they can use is either in the red or the green, and they've got lovely kind of labeling kits to do that with. So the kind of signal you kind of get is kind of initial states, then you get this temperature jump when you turn on the IR laser, you get then movement thermophoresis out of the spot, and then when you turn off the IR laser, then you get things coming back again, you get back to fusion. So that if, say, you had lots of capillaries and you had the same concentration of labeled material, and then you had different concentrations of an unlabeled binding partner, then you're going to change that thermophoretic movement because you change the size of the charge or the conformation or the hydration shell. And so you can fit then this binding curve, so it's a depletion curve. But often enough then, I think in another instrument that they have, and certainly you can do this with the MST, instead of actually looking at the total thermophoresis, you can just look at this initial temperature jump. And so this is called the temperature-related intensity change. And so just by looking at that, you can actually get a nice curves as well. And as you can imagine that, okay, so if you're changing the temperature, we saw with the Prometheus, and you're getting a decrease. But maybe if you then got something binding onto the protein, you're getting some kind of proximity effect around this fluorophore, or even maybe a conformational change. Right, and yeah, just as I said, then you kind of fit. Then again, with this is again a quadratic equation here, and that's just again a ligand depletion method. The reason why the MST is good, well, we've tried lots of different biomolecules, and it's worked for them. Vesicles, even ribosomes, DNA, RNA, everything. Of course, one component has to be labeled. So here's an example from the Hector group. And so they're working on the EMC complex. They got this cryo-EM structure, which of course was a bit, as you can see, they fitted in the kind of the crystal structure and of going, well, it's a bit and it's a bit and it's a bit, is this really real? Is the orientation right? Have they got these two things together? And so they then took two of the components they thought were in the complex, EMC2 and EC9. They then labeled the EC2, and then by MST then could find out what the binding constant is. So they're kind of sure that, yeah, this was really part of the complex. Right. So I mean, the nice thing about the MST is it's really very, very easy to use, and it can tell you whether your assay is going to work or not. So the downside, of course, is you have to label one of the components. So it's very important that you get one dye or less to the protein. Make sure you know what the concentration is, and correcting for the fluorophore absorbance at 280. You also need to use buffers, make sure they're nice, and you're going to get any scattering. Obviously, everything should be accurate. So you can do a kind of a pre-test in here. The software will kind of lead you through step by step. And it will kind of tell you, well, you're putting it into capillaries. What are the capillaries going to look like? That looks okay. Yeah, the intensity looks okay. It's the variation is good. You don't need any absorption. And then the MST traces, this is only for free samples. Looks, yeah, that's okay. So again, we're kind of going to use the fluorophore at a really low concentration below the KD. And you kind of need to check whether there is any absorption or whether there's any aggregation. So here's examples next. So if you get attachment then on the glass surface, there's quite a big, obviously, surface area relative to volume in these capillaries. And you get these little hats being formed. So you're getting a depletion of the fluorescence in the center and an increase at the sides there. So you can try different capillaries. You can put in detergents or change the buffer. And then when you get aggregation, you get these really horrible kind of wiggly rhythm traces. And what's happening there is that you've got a big aggregate, and then those are kind of just going into the field of view and coming out again. So again, you can try additives, detergents, BSA, PEG. Obviously check if you're going to do things. Check if you've got the MSO. You may need to then get your assay kind of better optimized by just changing the pH salt. And then maybe you could say, well, go back to the Prometheus, see if actually your protein is actually happy in the buffer that you've kind of chosen, or whether there's different ones. Also, you can use this kind of binding check in the MST before you kind of waste lots of proteins. Do a couple of samples at kind of low and high, and then it will kind of tell you, whether it's good or not. Kind of on the left-hand side, it's saying, well, okay, the variability of your binding is okay, and you're getting a good kind of signal window here. And then on the right-hand side, it's saying, ooh, it's all very noisy. So, you need to do some controls. So, easily we kind of set up a titration. You have a kind of a constant concentration of your label partner, just adding 1-to-1 to your unlabeled material, let it time for equilibrium. And we can just, do it for, this is a similar kind of thing you do for doing a polarization experiment. In this case, then you need to really ensure that your fluorescence is accurate across. This is kind of a good test of how good you are at pipetting. So you shouldn't really get much variability. Obviously, in the middle there, it's kind of like, ooh, it's a bit random. So is are things sticking, are things, is things not being very happy? And then on the right-hand side, then we can kind of see that, oh, we're getting a ligand-dependent change in fluorescence. Well, actually, maybe we can actually fit that. There we go, just before T. Further information, we've got a biophysics website, have a look at that. We can tell you all about different techniques. Here's all the different optical units we got for the kit reader. And we've got links to everything. And you can have a look at, what everybody else has kind of done, either by recently or actually then just by technique as well. Okay, and then of course you can go to the library and have a look at our book that me and that we edited. And for instance, you want to get into depth, then Lankovich is in the library as well. Okay, so thanks for coming back after the fire alarm. So is there any questions?